Okay. Hi everyone, we're looking at the effect of paraphenylenediamine on NAT2 gene expression, behavior, and L3 larva counts in Drosophila melanogaster. I'm Sydney Jones.
I'm Annie Galt.
And I'm Makayla Welch.
Okay, so Drosophila melanogaster is the common fruit fly. It's a eukaryotic organism, and it's great for these type of studies as it has a short lifespan and also a simple reproduction cycle. The larvae are large enough that we can perform mechanosensation assays on, which is what we did in this study. We focus on paraphenylenediamine, which is commonly called PPD. It's an arylamine compound. It's commonly found in permanent hair dye and cosmetics. In the 1970s, 2 arylamine compounds were removed from hair dyes because they had a small correlation with cancer with lab animals, but PPD is still in modern hair dyes today. We looked at the NAT2 gene in humans as it catalyzes the conversion of PPD to its monoacetyl and diacetyl metabolites, and it's found in the gastrointestinal tract and liver. We found some studies that showed that hairdressers and those who commonly got their hair dyed had a higher chance of bladder cancer, and that's why we focused on the NAT2 protein as it has the most association with bladder cancer. Our hypothesis is that the introduction of 3.4% PPD to Drosophila melanogaster will increase the expression of the NAT2 gene, will create less of a response to mechanosensation, and will have decreased number of third instar, which is the L3 larva, compared to Drosophila melanogaster that is just exposed to water.
Okay, so for the methods portion of this research, we got the NAT2 gene coding sequence using flybase. Once we got that coding sequence, we were then able to synthesize primers. We needed forward and reverse primers for the NAT2 gene, and these were designed using the Integrated DNA Technologies website, also known as IDT. So after we got our forward and reverse primers, we were able to start culturing our fruit flies. So the cultures were maintained in potato flakes with about 10 milliliters of deionized water and yeast pebbles as a food source. Once we cultured the flies, we separated them into experimental and control groups. The experimental group had 1.5 milliliters of a 3.4 solution of PPD dropped on top of the flies and the media, and that sat for a total of 24 hours. We did the exact same thing for the control group with 1.5 milliliters of deionized water, and that also sat for 24 hours. After the 24-hour time period, we ran a behavioral assay, and this is where we took the L3 larvae and they were subjected to petri dishes with PBS solution in them. The larvae were then sorted out and removed with forceps to be tested for any aversion to a poke in the thorax region. So this behavioral assay was all done by the same person so that there was no bias and we kind of had a baseline of what to expect, and so that our results weren't all over the place. And ultimately, the L3 larvae got a yes or no for a response to the poke, and if they moved in any way, if they wiggled their body, reversed, or kept moving forward, that was considered an aversion to the poke. So after we ran the behavioral assay, we then did a larva count of the L3s. So the larvae were extracted from the culture tubes, placed in the petri dish with the PBS solution, and they were sorted through and then counted. After the assay and the larva count, we then ran our RNA extraction. So the RNA was isolated from the L3s and it was purified with Keigan's RNase mini kit. We then performed reverse transcription on the RNA that was extracted from the L3s to make cDNA that was synthesized using RevertAid reverse transcription kit. We then performed qPCR, and we did this with the PowerUp Cyber, and we used the GAP-DH gene and its expression to normalize the expression of the NAT2 gene to see if we had any increases in the expression of the Drosophila that were exposed to PPD or those that were just exposed to the water. Now I'm going to talk about figures 1 through 3 really quick. This was just the setup of the experiment so you can see what was going on. So in figure 1, these are the petri dishes with the PBS solution and the L3 larva in the potato media. On the left is the is the Petri dish that had PPD in it. And as you can see, there's a discrepancy in the color when you compare it to the picture on the right, which just had water. So it kind of has that darkish color that dyed the media as well as dyed the larva and the living flies in there. In figure 2, this is a top view of our actual cultures before we put them in the Petri dishes. So on the top is the PPD and on the bottom is just the water. And once again, you can see that discrepancy in the color. The culture that was exposed to the PPD is much darker and actually dyed everything in there. And then the last picture on the right, figure 3, this is just a side-by-side view of the culture tubes as well as the Petri dishes after the whole experiment was ran. And this is just to get a better comparison of the changes in colors that we saw throughout this experiment due to the exposure to PPD.
For figures 4, 5, and 6, these were our data analysis results throughout the entire experiment. So figure 4 was focusing on the fold change in expression of the NAT2 gene between the Drosophila Melanogaster control group, which we just used water with, and that is bar blue, the blue bar, and the experimental group, which is the green bar that was exposed to the 3.4% solution of PPD for that 24-hour time period. For figure 5, this was our response fraction to the mechanosensation between, again, the control group, which was the blue bar, and the experimental group, that was the green. And then figure 6 is our data on the average number of L3 larvae that were identified during the 2 rounds of collecting this data over the course of the experiment. And again, the blue bar is the control group that we used water with, and the green bar is the experimental group with the 3.4% of PPD solution. Overall, in conclusion, our results showed a decrease in the expression of the NAT2 gene when they were exposed to the 3.4% solution of PPD. These are opposite to what we did hypothesize would happen. We hypothesized an increase, but we did show a decrease. Despite this, though, we cannot say strongly that these are significant results. Figure 4, that was only with 1 round of a qPCR due to the time and the capability of everything we had going on. We were only able to do 1 round. And so we cannot deem that significant because it has an N of less than 10. And then for figures 5 and 6, these both were also not significant due to the p-values being above 0.05. So to further the study and to make it more significant possibly in the future, we need more rounds of qPCR to be ran and have that N of sample number go up. We would also need to make sure we have a more stable environment due to everything going on with the world. We had less supplies, we had less time, and we had less ability to get into the lab and do what we needed to do. For future studies, I think it would be interesting to turn the focus from the NAT2 gene to the NAT1 gene, which is kind of the sister gene of NAT2. The reason we did NAT2 was because of the higher affinity for the toxins in the body, but NAT1 also does have an affinity for the toxins. So it'd be interesting if instead of using your focus on NAT2, we could turn the focus to the NAT1 gene and see if this could give more insight to the PPD's effect on the human body.
We would like to give a special thank you to our advisor, Dr. Stephanie Ottohit. Without all of her advice and critiques, this would not have happened, and especially with everything that's happening in the world today, without her dedication, we would never have been able to finish this. So thank you. And then thank you to everyone for coming to our presentation today.
Thank you.